Thank you all very much for coming. The first thing I'm going to do is introduce our speaker. Mike Piehler is a joint appointment at UNC Chapel Hill, where he is a professor at the Institute of Marine Sciences and is also the program head in Estuarine, Ecology, and Human Health here at Coastal Studies Institute. Hello, thank you. I am Mike Piehler. So tonight I'm going to start off with my slightly showy title. In the holiday season, we're competing for everyone's interest. And so I'm going to be talking to you about our research on oyster reefs. And this is actually a sequel, so it has the added challenge of trying to get attention after having something a little bit similar presented about 3 years ago. So my title tonight is On a Highway to Shell, Assessing Oyster Reef Function Throughout the Southeastern United States. So I'm going to walk you through a bunch of field work and experiments that a group of us did in a project funded by National Science Foundation that was trying to better understand how oyster reefs function throughout, intertidal oyster reefs function throughout a large part of their biogeographic range. So this goes from about the middle of North Carolina to about the middle of Florida and includes a little bit of the Atlantic coast, mostly Atlantic coast, and a little bit of the Gulf Coast. So in case any of you weren't here for the first installment, Episode 1 of the oyster trilogy I've decided it's going to be was Ecologically Valuable and Delicious, the Paradox of the Eastern Oyster. So what were the main points of that talk? One of the main points was this fact that through a number of factors, including habitat degradation, overfishing, loss of population sources, we've lost oyster reefs throughout a lot of the United States and throughout the world, as you can see. And this is problematic for a lot of reasons. People love to eat oysters. Oysters are an important fishery in a lot of places. And we also will talk more tonight, and I did in this talk, about all the valuable things that oyster reefs do that we don't necessarily know about. These things include the obvious production of oysters, perhaps obvious to most, the augmentation of fish production, so creating habitat that allows more fish to grow. Water filtration, obviously oysters take things out of the water and put them into the sediments, and this has effects on the clarity of water, and it also has effects on the quality of water. They stabilize the sediments and adjacent habitats. So if you have an oyster reef at the edge of a salt marsh, that salt marsh is likelier to sustain itself in the long term. They provide habitats for invertebrates, small organisms that not many people think about but are important for the productivity of the overall system. They also potentially sequester carbon by fixing carbon and putting it into the sediments and burying it. And then finally, they diversify the seascape, which is an important feature of habitat function. So one of the take-home messages of the last talk was that all of these services have economic values. And this paper by John Grabowski et al. That Andy Keeler and I were co-authors on quantified the values of these ecosystem services. And one of the interesting things we found was that the red large part of the pie, the nitrogen removal part of the pie, was larger than we might have anticipated. A significant proportion of the total value of these reefs was associated with that nitrogen removal. And this was interesting and great, however, it was fairly geographically focused. So for this topic and for some of the others in this paper in terms of services that were evaluated, it became clear that knowing something more about how these things vary on a broader geographic scale was important. And better still, hopefully knowing something more about why they vary on a broader geographic scale would be great. So that's what I'm going to talk to you about tonight. There's some more details about field and lab work that we've done to try and get at, do these things vary across a geographic scale? And if so, why might that be? So one approach we could take is to head out down the coast and walk to all the oyster reefs along the Atlantic seaboard. I calculated on Google Maps that that would take 8 days of walking without stopping, so that is not an option. We had to find another way. And so we were fortunate to be working as a part of this great team that included the aforementioned John Grabowski, who's at Northeastern University. I was a part of the group, Jeb Byers, who's at University of Georgia, and David Kimbrough and Randall Hughes, who were then in Florida and who are now at Northeastern University. And so with this group, we were able to take on this broad geographic assessment. Each of these dots that has a label are different sample sites in the northern, central, and southern areas. And I'll refer to this northern, central, and southern regions throughout the talk. So perhaps I'll keep showing you this map, but you can retain this image in your brain. So John and I were in charge of the northern sites, which were in North Carolina. Jeb took care of South Carolina and Georgia, and Randall and David were in charge of Florida. So because we decided that walking was impractical, we all got in our state-owned vehicles and headed out on the highway to Shell, which turned out to be Interstate 95. For the most part, took us to all of the oyster reefs that we needed to get to. So this was a lot of driving and a lot of work, and there's a reason that we don't have these big, broad geographic studies, because they're really difficult and really time-consuming. So we feel very fortunate to have had the chance to do this work. So there are a number of things that oysters do. I've already run through an overview of the ecosystem services, but they do a lot of things that modify the sediments. Just by existing as structure in the water column, they baffle the water and allow things to settle out, which tends to increase water clarity and tends to increase the accumulation of things around them, including sediment organic matter, which is an important part of organic matter, an important attribute of sediment. They also, through their filtration, bring lots of things to the sediments that wouldn't be there otherwise, including algae and suspended particles. So in doing that, they change the light environment. There tends to be, following their filtration activity, there tends to be more light available, and that increases the ability of photosynthetic things to grow at the surface of the sediments. And those include submerged aquatic vegetation, or seagrass, but it also includes benthic microalgae, who are less appreciated but also very important benthic primary producers. So as a part of our big biogeographic effort, we did a big sediment sampling campaign. And a lot of this was driven by this connection that we had seen between this loss of the oysters that I talked about in Mike Beck's paper early on, and converting that to a loss of oyster filtration. So people have talked a lot about nutrient management requiring source reduction, and that's certainly the case. But nutrient management may also benefit from increasing places that remove nitrogen. And oyster reefs, we know from our early work, are one of those places. So in our efforts to feed the global population, you can see that this figure in blue has global population growing into present day. It also has increases in total reactive nitrogen. And that's a result of our becoming very good at fixing atmospheric nitrogen into fertilizer. Following World War II, the commercialization of the Haber-Bosch process allowed us to get nitrogen from the air, literally, and made us much more effective at growing crops to feed the world's population. So this is not, on the surface, a bad thing at all. However, unfortunately, it was done fairly inefficiently and increased the amount of nitrogen available throughout terrestrial and aquatic environments, and created situations in estuaries in particular where there was too much nitrogen and there were things like algal blooms. So corresponding with that, there was this loss in the oyster population that the Beck paper talked about. And this takes some oyster data from Filene Zuermgassen's paper in 2013 and converts them to filtration capacity. And what you'll see is that with the one exception, which is Apalachicola Bay, which is actually a place that has lost most of its oyster filtration, these data were just taken at a time where there happened to be quite a few more oysters than there are now. But for the most part, you've seen nearly 100% loss of oyster filtration capacity. So this part of the function of estuarine ecosystems is gone. And so many people have thought if we could restore this function, we might be able to do something to gain traction in the overall efforts to improve water quality in estuaries and to increase to decrease nutrient levels and increase water quality. So off we go. We're headed out and we're taking samples throughout this giant geographic range. These are the names of our sites, here indicated by X's. And we visited them 4 times during the first year to get a sense of the seasonal patterns and all of these things. I apologize for the small captions here, but what's most important in this figure is just to show you a ton of data. And so the top panel is the north region, the middle is the mid, the bottom is the south. These bars represent concentrations of chlorophyll A, which is a proxy for algal biomass. So these are benthic microalgae, the algae that live on the sediments. When you see them on the sediments, they're either gold or green, depending which ones they are. And they turn out, for reasons I'll talk about in a minute, to be really important for ecosystem function. And so really the take-home message here is that we had lots and lots of data. The light bars are on reef, the dark bars are off reef. So this is comparing areas with and without oyster reefs. And what you see is a general pattern of enhanced biomass on oyster reefs and lower biomass off oyster reefs. But as you go from north to south, that enhancement is a little greater and that difference is a little greater. So we summarize it with this figure, which shows this is the mean, so the average of all of those bars. So the top panel is now the left-hand side, the north. The middle is the mid again, and the right-hand side is the south. Again, light bars are on reef, dark bars are off reef. And there are two really striking things. One is that the on reef, the dark bars, as you go south, the biomass of benthic microalgae increases. The other thing that was very striking and interesting to us was that the off reef, the comparison site, the control, if you will, the light gray bars, don't increase as you go south. So as you go south, it's getting warmer, you're having more light available, all the things that you would think would benefit organisms that photosynthesize. But it turns out there's probably something else limiting their ability to accumulate biomass. And in this case, it's likely nutrient limitation because the on-reef sediments have much higher nutrient concentrations. So the activity of these benthic microalgae, or the biomass of these benthic microalgae is very different, and I'll talk in a second about why we think that might be important. But it's also important to know who these algae are. If they're nuisance algae, this goes from being potentially an ecosystem service, a good thing, to being a problem, and we certainly want to know if that's the case. So we did analysis of the photopigments using a high-performance liquid chromatograph. Matt Waters did this analysis for us. And so we looked at chlorophyll A, which I've already talked about, which is the indicator for algal biomass. But we also looked at fucoxanthin, which is the indicator for diatoms. And diatoms are very palatable. They are almost never nuisance organisms when they're associated with sediments. And so it was good news that most of the augmentation of algae was diatoms. The bottom right panel is intentionally left blank because there were no other pigments present. Virtually all of the pigments that were present in the system the chlorophyll was represented by the fucoxanthin. So why are diatoms important, or why are benthic microalgae important and diatoms in particular a good thing? I mentioned this already. They're very important at the base of the food web. So what that means is they're sort of the grass to support the grazing animals. What we know is that if you're in a salt marsh, for example, a very small proportion of the total salt marsh biomass ends up in a food web. But a tremendous proportion of the benthic microalgae, up to 80%, ends up in the food web. So these are great food. So having sediments on oyster reefs with augmented sources of these benthic microalgae is a great thing for helping grow everything else that's on the reef. And this is part of the reason why they are a great habitat. Additionally, there's something called the benthic filter. And these two schematics show pictures of what it looks like in the top panel, where if you have benthic microalgae living on the sediments, you're less likely to have nutrients coming out of the sediments. So it's effectively capping the sediments and removing an internal source of nutrients. And we talked about controlling sources, controlling sinks as two parts of nutrient management. This is another way to control an internal source of nutrients that can go a long way to helping you get a handle on nutrient enrichment problems. On the other hand, in the bottom panel, when you don't have benthic microalgae and you don't have benthic primary producers, you tend to have faster turnover of nutrients in the water column, and you tend to grow phytoplankton, which are often toxic. So these are algae living in the water. So everything goes quicker. You tend to have nuisance organisms. You tend to have organisms that are potentially harmful or toxic. And so by far, the top panel is the more desirable. But you can quickly see that these have feedback loops, so that as you begin to develop algae in the water column, you tend to quickly lose the benthic primary producers in the top panel. So maintaining this balance is important, and oyster reefs appear to make a contribution to helping foster the benthic microalgae. So here's a picture of them not looking quite as glorious as they did in one of the versions that I had, but you can see the golden sheen, which is the diatoms. So we go back to our study sites, just to remind you of where we're going. And this time we're going to look at a different attribute of the sediments, organic matter, which is one that I mentioned. Organic matter is really important because it controls a lot of things about productivity in the system. What types of organisms live in the sediments. It has a direct correlation with the nutrients available for plants to grow. So this is the paradox of nutrients that I think probably most of you have thought about, is that you need some for things to grow. You just don't need too much for the wrong things to grow. So it's not an issue of trying to manage something away. It's trying to manage something in moderation, which is a bigger challenge, much more so than just eliminating something. So we're interested in how oysters modify sediment organic matter. Sediment organic matter has been shown to be closely connected to nitrogen cycling, which I talked a little about and I'll talk more about in a second. And it's also a really good indicator of the status of the system. So again, this figure is mostly to show you that this is tons and tons of data from lots of different places. We're going from north to south, so Middlemarsh around the corner, Middlemarsh in North Carolina, around the corner to the Gulf. What you see if you look at these pairs, which are reference and oyster sediments respectively, that generally speaking, the oyster reefs augment sediment organic matter relative to their control. If you look in the middle of the distribution, starting around North Inlet, NI, and going maybe through Sapelo, that pattern starts to break down a little bit. And so if you look at this summary, again, of the north, central, and south regions, you'll see that there's some augmentation of organic matter by the oyster reefs in the north. There's a lot in the south, but there's not too much in the central. And Jeb Byers, one of the members of our group, published a paper a couple years ago where he looked at the effect of tidal range on all the things that we see in the oyster reefs we worked on. And you can see that that central part of the range has the highest tides. And so these are isoplus of tide. And so you see you have up to about a 3-foot tide in the middle of our distribution, as opposed to something like a 2 and sometimes a 1-foot tide in the outer areas. So these areas with the higher tides are not showing an augmentation in organic matter as a result of what the oysters do. And there are several reasons why this might be. If you have a higher tide every single tidal cycle and you have organic matter in the water, it's going to come in, fill up at high tide, and settle out. So it may very well be that a lot of the organic matter is being delivered by these higher tides and swamping the signal that we see in the oyster reefs. It was interesting, though, in that paper of Jeb's that I mentioned, now we're going from low latitude to high latitude, so from south to north, looking in the top panel at live oysters, the middle at oyster cluster weight, and the bottom at a recruitment per reef, so oysters coming into the reef. And generally speaking, everywhere along the way in the middle part of the distribution had bigger oyster reefs that recruited more, that had larger cluster weights. So if you were going to look at this and say, okay, which part of the distribution, which part of the geographic distribution is going to have the most augmentation of organic matter, you would guess the central part of the distribution. It turns out to be absolutely the opposite. So what we did was look at the difference in on-reef and off-reef. So this is oyster augmentation, and we compared that to oyster density. And we did so in the blue, which is the south, the sort of pinky purple, which is the central, and then the yellow, which is the north. And we looked at this relationship between oyster augmentation of organic matter and oyster density. So if you look at the blue and the yellow, particularly the blue, which is the southern area of the Florida sites, as you get more oysters, you tend to get more organic matter augmentation. And pretty much the same is true for North Carolina, just less augmentation. But if you look at Georgia and South Carolina, no matter how many oysters you get, you have the same amount of augmentation, and it's very little. So this is speaking to something physical overwhelming this biological activity. So we're not seeing the oyster signal at all, and perhaps even the higher tides are contributing to smearing away any organic matter that the oysters bring into the sediments. So this looks like it's something pretty different than that which we've seen in most of the work we've done in North Carolina. And it may have important implications for the nutrient cycling stuff that I'll talk about in a minute. So on that topic, we take a great picture of an oyster reef and lay the nitrogen cycle on top of it, which I'm not going to go through in any detail. But I do just want to orient you to thinking about this idea of the nitrogen cycle. I like this representation because it looks very stable and it looks very balanced, and things cycle through. You start off with inorganic and organic forms that go through, come around to nitrification, which creates nitrate, which is biologically available. Some amount of that is denitrified, and then you can come back around starting at the top at N2 and have a biologically mediated process that brings nitrogen back into the system, nitrogen fixation. And that's what the industrialization of Haber-Bosch has really increased that nitrogen fixation term artificially. So you have much, much more nitrogen coming in than we once did. So to balance that, we need to figure out a way to have as much or more, or as much or at least more denitrification. So we did a series of experiments, again in the north, in Georgia this time, not in both Georgia and South Carolina and in Florida, in which we compared reference sites, so away from an oyster reef, and oyster sites. So these are taking sediment cores from on oyster reefs and off oyster reefs, running these incubations we run, and coming up with this rate for denitrification. So this is the nitrogen removal, the left side of the nitrogen cycle there. And what was interesting, if you look at just oyster and follow that down, so the second bar down and then the fourth and the sixth, as you go south, you get more denitrification on oyster reefs. But the difference between the reefs, again, is really similar to what we saw in the organic matter pattern. So there is some difference in North Carolina, a pretty big difference in Florida, and not much at all in Georgia. So likely, again, we think it's attributable to some physical feature creating larger organic matter pools in all sediments rather than just on the oyster reefs. So we knew going into this project that predators played a big role in oyster reef function. This is a picture that I staged. I found a blue crab claw, and I decided that it represented predators interacting with oyster reefs. And so it's in great focus, but perhaps a little bit out of context. But I use it to remember to begin talking again about the whole of the reef. So we think about oysters as filtering machines, and doing so, we oversimplify oyster reefs and probably will misunderstand their function. I've already talked to you about several things about oyster reef function that we wouldn't have predicted if oysters were just simply a filtering machine. But because they have predators, oysters have predators, they have a whole suite of crabs that live on them, they have all these trophic interactions that go on the oyster reefs that affect oyster reef function. It's really important, if you're going to make broad generalizations about what you expect from oyster reef function, that you have a good understanding of what's driving it. So again, this process, denitrification, we conducted some experiments in which we had oysters alone. So these are the red bars. The blue bars are mesopredators. So these are mud crabs that I'll talk more about in a second. Mud crabs eat oysters. And then predators. Those are either blue crabs or toadfish who eat mud crabs. So this is a complete food web. I'm going to show it to you in just a second in a graphic representation. But what we were trying to understand is, how do predators affect the process of denitrification? The original data that we gathered were from Florida, and so it looked really promising that we were having these effects of predators that were depressing oyster-mediated denitrification. But it turns out, as we gathered more data, Georgia and North Carolina, it didn't make a ton of sense for this particular process. Again, though, that similar pattern of increasing rates of denitrification as you go from north to south. We also measured the flux of ammonium. So this ammonium in the nitrogen cycle is one of the really biologically available forms. This is important because if you're going to suggest that oyster reefs are good for nutrient cycling because they remove nitrogen, you have to know the degree to which they're a source of nitrogen. And ammonia is a really biologically available form, and so knowing this is really important to a rigorous assessment of the degree to which oysters are beneficial for ecosystems. So if you look at the oysters alone in both North Carolina and Florida, they had pretty big ammonia fluxes. But interestingly, when you had any crabs at all, whether it be mud crabs or blue crabs, that ammonia flux went down. So this is something that is increasingly actually a topic for lots of different systems, is understanding the role of larger organisms and understanding the role of predators in modulating nutrient cycling impacts from places like oyster reefs. And so what we know from this is that if we had an oyster reef and it didn't have crabs on it, there would be a balance between nitrogen removal through denitrification and then this significant flux of ammonium coming out. But when you do have the predators present, the crabs present, you don't see that. And so that increases something that's called your denitrification efficiency. And so this is a way that you can measure the degree to which this habitat is effectively removing nitrogen rather than creating new sources through the ammonia we talked about in the last panel. So all of this is to say that the more we understand on a broader geographic range about the function of these oyster reefs, the certainty with which we can say whether or not it's a good thing increases for sure. And so we wanted to know in more detail how these predators were affecting nitrogen biogeochemistry, which is what I've just been talking about. We know that many of them directly consume oysters. We know that they can modify oyster behavior, including decreasing their filtration. They are responsible for bioturbation, which is digging up the sediments or just aerating the sediments and changing sediment properties. And then finally, they potentially could be scavenging materials from the sediment surface, which could change the way that nutrient cycling goes on. So we know from John Grabowski's early work in 2004 that these mud crabs that I've been talking about have negative effects on oysters. They eat them, and they also scare them, and they change the way that they filter. And from John's work, we also know that predators, like this oyster toadfish and also blue crabs, when they fit into this food web, they have negative effects on the mud crabs, which can have an indirect positive effect on the oysters. So you can imagine the toadfish scaring the mud crabs makes things better for the oysters. That makes sense? So John showed this unequivocally and convincingly in experiments and in field experiments in North Carolina, but we wanted to know more about this and the degree to which this was transferable across the geographic range. And so this, as a part of our big data gathering effort, same team, same areas, but this time doing things related to oyster reef ecology. So looking at oysters, small crabs, and snails, catching things in gill nets to figure out what fish are there, including transient fish, and then monitoring environmental conditions to try and understand how things varied and, importantly, why things varied. So in the meantime, with the first set of data that we gathered, David Kimbrough, part of our group, published a great paper in Ecology Letters that looked at this interaction between the predators and the mesopredators, the mud crabs, and the oysters. And what he found was that not only was the magnitude of the effect different as you went from North Carolina to Florida, so you have a decreasing effect, you go from a positive effect, which is the one that we talked about, where the predator either eats these or scares these, which has a benefit for the oysters. And in fact, as you go south, you see negative effects. And it turns out, because life is never simple, the mud crabs do eat the oysters, but they also walk around on the surface of the oyster reefs and push sediment off, which is really beneficial to oyster reefs. So again, another example of the more you know, the less you understand, or the more you understand and the more wrong answers you figured out you had. So we then went to look, David took this on and looked at all of the data we had, and using structural equation models, tried to figure out what was driving the things that we saw. So we see these patterns in oyster recruitment and oyster reef biomass and mud crabs that I showed you from Jeb's paper, and it turns out that water depth is the primary driver. So for the reasons that I talked about, every single tidal cycle, more water is coming in. It's bringing in more potential oyster recruits that increase the biomass of the reef and thus lead to more mud crabs. And so if you look at the range in latitude again, going from south to north, looking again at the figure from Jeb's paper, this is supported by oyster recruitment. So the areas where the tides are the highest, we saw the highest oyster recruitment by far. And the areas where they're lower, we saw lower oyster recruitment. But this is interesting because this water depth also applied to piscivorous fish and, interestingly, to me at least, to sharks. So in Georgia and South Carolina, the tides are high enough and the water is deep enough that bonnethead sharks have access to these reefs, which struck me at the time as someone who doesn't know anything about this as pretty neat. Well, it turns out it's pretty neat for sure, but it's also really important because these bonnethead sharks are only present in part of, these are intertidal oyster reefs, so the water rises and falls each tidal cycle, fills them up and then drains off. And at high tide, you have these bonnethead sharks, and they're non-trivial, they're big sharks. And that is at once interesting, but it's also important for function because bonnethead sharks, it turns out, like to eat blue crabs, who are one of the important predators who eat and change the behavior of the mud crabs. So we have a really different food web in the central area. So the central area has higher tides, which means that more material is delivered to the oyster reefs every single tidal cycle, but it also has higher tides, which brings deeper water and totally changes the food webs. So from this, what do we know? And really, what does it mean? What information can we glean from this that is useful? So we know that sediment modification varied geographically and that it wasn't consistent for different metrics. So sediment organic matter, augmentation by oyster reefs was not present in the central part of the distribution, but was really prominent in the southern. Benthic microalgae, which we know are also important, were augmented the most in the south and mostly varied with latitude, whereas most other things did not. We also know that food web structure changed. And for all the reasons we talked about, there were differences in water depths. There were differences in some temperature differences that we're looking at more now, but the tidal range was the main thing. And so these things combined to make reef function vary significantly throughout the geographic range. And importantly, this probably has significant implications for our understanding of the ecosystems and for ecosystem services. So I didn't define it in the beginning, and I apologize, but ecosystem services are the things that natural systems do that are of value to people. So they necessarily have to have a value somewhere. And so in the case of an oyster reef, harvest is one ecosystem service. Certainly water quality improvement can be. But I hope you can see from these data that certain services like water quality improvement are going to vary really dramatically throughout the geographic range. It looks like there's probably not a giant denitrification enhancement in Georgia and South Carolina. So if you're going to do a reef restoration, or if you're going to do a living shoreline, and part of your success is gauged on having that benefit from water quality improvement, it'll be important to make sure that you're convinced that that's the case, because our results suggest that there may not be as much of a potential for water quality improvement in those areas with the higher tides. Similarly, in southern areas, it appears that all of the augmentations, the benthic microalgal augmentations, the increase in organic matter are much higher. And that's likely due to a lot of factors, but probably has a lot to do with the consistently warmer conditions. So you may get more return in those areas. But again, the more we look, the more we were surprised by what we found. So all of this we think about in the context of oyster reef function for certain, but there are lots of applications to restoration, as I've already talked about. Increasingly, oysters are a part of many of our environmental solutions. So people talk about living shorelines. Oyster reefs very often are included in living shorelines. And so if we're going to use these things, people talk about nature-based infrastructure, and that sounds great and it's very appealing. But if you're going to build infrastructure based on nature, I hope you are convinced now that it's critically important to understand how they function in nature and how that varies as you look in different areas. So we're excited to be wrapping up our field work on this project and continuing to develop products. And hopefully this is the second of 3 talks perhaps on this topic. Maybe I'll choose. The first of them was in competition with the jam band Blues Traveler, and the second now is Christmas time, but I appreciate you all coming. With that, I thank all of my collaborators. I went with the youthful pictures of all of us and thought that was a funny haircut, but then looked in the mirror and realized I have almost the same haircut. And John Grabowski and Randall Hughes building an oyster reef, David Kimbrough, my students Ashley and Kathleen, who've been instrumental, and Luke Dodd, and Suzanne Thompson, lab manager in my group. And with that, I will thank our funding sources. North Carolina Sea Grant funded our first project on this topic, and since then it's been supported generously by NOAA National Science Foundation, and I would be happy to take any questions. Thank you.